Okay, so I'm doing drug targets for the CSF1R mutants investigated by high throughput virtual screenings of zinc 15 library compounds. So a lot of my data was based off of the bioinformatics student. So colony stimulating factor 1 receptor, which is CSF1R, and that's what I'm going to be talking about the whole time, is a class 3 tyrosine receptor kinase that is involved in macrophage chemotaxis and differentiation. It is classified as a proto-oncogene. So it has established regulatory roles in a range of cancers, including lymphoma and acute myeloid leukemia. However, it is most commonly associated with tenosynovial giant cell tumors, which is TGCT. So they are a rare type of tumor that affects the synovial tissue lining, joints, and tendons, and can cause pain, swelling, and stiffness. The pexidartinib is an FDA-approved drug, and that targets the CSF1R, and it has shown promise in treating the TGCTs. However, the response varies, and not all patients will benefit from this therapy. So therefore, the study is going to aim to investigate the relationship between the CSF1R mutations and the response to pexidartinib therapy in TGCTs. Okay, so the primary objective of the study is to use high throughput virtual screening of the zinc 15 library and identify potential novel drug targets for these mutants. So the specific objectives are to identify compounds that bind to the active site of these mutants using virtual screening, and then we're going to investigate the effect of the wild type and mutant. I just realized that I didn't include the mutants in this, but it is in my paper. And then we're going to analyze the binding modes of the identified compounds using molecular dynamics simulations. And then we're going to validate the biological activity of the identified compounds using experimental methods. We used all the methods that we basically went over. So like the protein database, and then the docking, and then the molecular screening, all that. Actually right here. So the first one, this method involves searching the protein data bank for available structures of the wild type and the mutant type of CSF1R. And then the protein structural properties. So this method involves analyzing the structural properties of the wild type and mutant type of CSF1R, including solvent accessible surface area, secondary structure, and hydrogen bonding patterns. The modeling method involves building a three-dimensional model of the mutant type of CSF1R based on the known structure of the wild type protein, and then docking involves predicting the binding mode and affinity of ligands to the active site of the wild type and mutant type of CSF1R. And then lastly, the virtual screening will use the computational methods to screen a large library of compounds to identify potential inhibitors of the wild type and mutant type of CSF1R. So innovative aspects. This proposed study is original and innovative because it aims to identify novel targets for therapy based on the downstream effectors of CSF1R signaling. And then this approach has not been widely used in the field of cancer research. And it also has the potential to identify new targets for therapy that are more effective than existing inhibitors. It provides new insights into the molecular mechanisms that drive the response to pexidartinib therapy in TGCTs. And it also provides potential biomarkers for patient selection for pexidartinib therapy. So what we're expecting in these results is an identification of cancer types with high expression levels of CSF1R, determination of the effect of CSF1R inhibition on cancer cells proliferation and survival, and then identification of downstream effectors of CSF1R signaling in cancer cells, identification of novel targets for therapy based on the downstream effectors of CSF1R, and then overall, this proposed study has the potential to identify the new targets for therapy that could improve the outcome for cancer patients. And that is all. Yes. They are in my paper, and I realized that I didn't put them in here, but I don't know them off the top of my head. I know them, but like I didn't put them in here. They're in my paper though, don't worry. But this was just a bunch of words, so I didn't have any papers.
Okay, so TGCT. Where is this tumor body?
That is actually a great question. Well, I know. Well, I said that yeah, it does affect the joints and tendons. Like,
And okay, the design portion you want to present will be. So you have 5 compound plus the this drugs of 6 compound. Then you have the wild type mutant. So then you have to have the 12 experiments found out the 12 IC50. For each kind of IC50, you have to have 5 concentration. Right? You have the other control concentration. 3 inhibit concentration. And if you have IC50, that easy. Don't have IC50. Use some high iron. Those details are important because it's a proposal. That's proposal. So you want to work. Also, the application of the talk, we explain some the IC50 for the mutant should be smaller. Smaller means important. Mutant isn't draw. For the wild type, actually we hope we have larger IC50 because we don't want targeting on wild type. That putting in your discussion, expect the results that such a compound to identify.
Right.
So the middle part.
A little bit more? Okay.